You're listening to the Knowledge Archives podcast. Welcome to the Knowledge Archives podcast. We're a group of students on a mission to learn from as many different disciplines of knowledge as possible. I'm your host, Madhav Malhotra, and today I'm glad to be joined by Dr. Manuela Nagel, Head of Cryo and Stress Biology at the Leibniz Institute of Plant Genetics and Crop Plant Research in Germany. Her work focuses on the long-term preservation of plants' genetics, specifically using cryopreservation for things like gene banks. So thank you for joining me today. I really appreciate you taking the time, and I'm very excited to learn more about this very futuristic area of research. And just before we dive into all the details, I'd love to hear a little bit more about yourself, your story, and how you got started in this area of work.
Yeah, hello, my name is Manuela Nagel. Thank you very much for this invitation to your podcast. I studied agriculture in Germany and I was quickly fascinated by gene banks, actually by the maintenance of plant genetic resources. And therefore, I came here to Gartersleben to do my master's thesis, actually. And then it somehow developed on its own. So finally, I continued with my PhD thesis with a postdoc, and now I'm responsible here for the cryopreservation unit of the Gartersleben Gene Bank. So that's the federally ex situ gene bank for agriculture and horticultural crops in Germany.
Yeah, I think it's really incredible to hear about that background and especially given this area. I know you immediately were diving into some of the details like the prominence of gene banks in the agricultural industry, etc. And I'd love to talk a little bit more about that. Just before we get started, though, I would very much appreciate a clarification of what are these terms like cryopreservation, like gene banks, and how do they work in a real life context?
I would like to go a bit back to the history. So, at the beginning of the 20th century, actually, there were a lot of researchers who started to realize that our plant genetic resources vanish. And this means that, there were farmers who developed our wheat and barley over centuries. And in the 19th century, with industrialization, the land races started to vanish. So, because we focus more on breeding tools, and so some researchers realized this and started to collect these land traces. So, and the first one who really did this systematically was Nikolai Vavilov, and he put that in the first gene bank, he called it, in St. Petersburg. And at the same time, also in the US and everywhere in the world, people realized this phenomenon and started to collect seeds around the world because they know that they need this resource for their breeding in future, for example, and for the maintenance of really valuable resources. And this is how it came to gene bank. Yeah, in the gene bank, usually more than 90% of this material is stored in terms of seeds. And as you refer to different terms, so we have a gene bank and we have a botanical gardens, for example. They do the same, but somehow in a different way. So a gene bank is really responsible to maintain the genetic diversity within the species, for example, a lot of different weeds, whereas the botanical gardens, they focus on the maintenance of species. They would like to have a lot of different species, but do not focus so much on the diversity within the species. So that's there the difference. So now we came to the cryopreservation. Over the time, of course, we realized that not all plants can be preserved by seeds because some of them do not either produce seeds, some of them produce seeds, but they produce seeds which cannot be stored. So that are so-called recalcitrant seeds. So they do not want to be dried that much and also doesn't like cold temperature, for example. For those kind of species, we need a different way. Therefore, people started to establish so-called field gene banks. So we just kept them in one place, but together outside still in the field. But the problem is, of course, in the field, you have sometimes different weather conditions. Sometimes you have material from all over the world and they don't like the specific location. So there is a strong risk that you lose these accessions in a field gene banks. And therefore, in the 80s, there is the International Board for Plant Genetic Resources, especially they tried to collect the ideas and started to set up so-called in vitro banks. So that was a medium-term storage. So we took the plants from the field and put it in tissue culture and tried to have a kind of backup there. So with tissue culture, you have controlled conditions. It's a bit easier sometimes to maintain it, but you have a lot of work. So, and since the 90s, the Japanese researcher Sakai, he started and had developed a method to easily bring plants into cryopreservation. And that was the point that we moved from tissue culture into cryopreservation. And here you have the storage of plant tissue under ultra low temperature, so at minus 197 degrees in liquid nitrogen. And this approach is then usually the long-term storage approach for plants which cannot be maintained as seeds. So I think now I gave a broad overview of all terms and the connections between them.
Yeah, I think that's really useful, and I think it's amazing to hear your knowledge of this historical context because it's really, really useful to understand how this field might progress and what some of the key objectives that people wanted to focus on over time are. And one final thing regarding this introductory bit is it's usually that we want to maintain genetic diversity because of agricultural applications like crossbreeding, etc. Are there any other main applications that we want to use this kind of technology for?
For us, of course, I'm working in a gene bank, so the main target is to maintain genetic resources over long term because for this vegetative propagated crops, it's really the only approach you can use to maintain the species. So all other approaches are more risky. However, of course, there are also different other approaches you can use from cryopreservation. There's, for example, the cryotherapy. That means that you cut, now you must imagine you have a plant and you grow them in vitro. That means you grow them on artificial media in a box with light. All conditions are set, so it's perfectly. And now from this plant, you take the buds, for example, the meristematic tissue, and try to cut that, excise this on a very small level. So it's very the basic level of a plant. And if you do that, you have a big chance that you can get rid of a lot of viruses and bacteria. And this approach is used to rejuvenate plant material and get rid of all this bacteria because they accumulate over lifetime of a plant. And there's also actually not many other approaches. I mean, you can also use chemical, but usually all approaches available are used together. Cryotherapy in combination with chemical and also heat treatment to get rid of all these microbes. Therefore, it's also applied. Otherwise, for cryopreservation, you have a lot of different areas, of course. I mean, in humans, it's used to store blood, for example, to store cells. Also for animals, it's the same direction, or to store sperms or embryos for humans, or, well, there is a broad range, but all have a similar task. The only difference is sometimes the question, does the material needs to stay viable? So, I mean, for us, it's very important that we have viable tissue at the end, because after cryopreservation, the plant needs to go back and needs to start metabolizing again. But sometimes in different areas, you just want to keep this material somehow in the best condition. So there, it's not important that it's alive.
Yes, and I think that is now a good time to transition into some of the details of how we have a few challenges in this approach and how we get around them. Now, I was looking through some of your research papers and I found keywords that I would love to learn more about, but I don't entirely understand. Like vitrification that I've heard about in other contexts, like cryogenics for humans. But I was very curious about how these processes can somehow stop the biological processes of plants. Like you mentioned metabolism as well. How do we go from stopping those to then making sure that the plants are safe from environmental conditions, oxidation, etc., and then get them back out of that state?
I start from somehow from the beginning. So to be able to maintain plant material, I think the processes are in nature everywhere the same. To have somehow a long-term maintenance, all the plants started to establish a system which goes back to this vitrification approach. So you can compare seeds with resurrection plants and with cryopreservation, which is then more the artificial way to get the status. And they all do vitrify to bring the tissue in a level where it's not freezing, it's just stopping from one to another moment. And seeds, the vitrification is the drying. So you take out the water and then all the membranes adjust and all the proteins start to develop in a way that they go between the membranes and stabilize the whole system. And this is the same also in resurrection plants. This is known. And they are able, as soon as water gets in, that they develop back and can just metabolize. And we try to take this idea of stabilizing the whole system and try to transfer it into the cryopreservation approach. So this brings up a huge problem. So you cannot do this with all the species, especially species which have extremely high water content, are a huge challenge where the membranes are in a different way. That's also a huge challenge, but you know already from my terms, we know not all about that, what affects really the cryopreservation success. However, I tell you now a system which works. For example, we would like to preserve potato because potato is one of the plants where most genetic resources are actually cryopreserved. I think we have more than 5,000 different accessions in cryo, and this is especially in Peru, in the US, and here in Garterslehm, that are the big centers for potato cryopreservation. And what we do is we grow the plant in vitro, then we just take the upper shoot tip of this plant, cut it again down to 1-2 millimeter size, including the meristem and maybe 1 or 2 leaves around which somehow protect this meristem. And then we bring it into a sugar solution. I forgot one point. We grow in vitro plants already under low temperature for a certain type that they somehow acclimatize. So they are a bit cold adapt, so at 5 degrees. So then we do the excision and then they go into a sugar solution. And at this step, already the first water is taken out. So this reminds you now on this vitrification approach. Yeah, and we start to get the water out. And also the sugar might somehow diffuse in, but this process is probably not so strong because the sugar molecules are very big. So it's hardly that they go quickly, and if they go very slowly. And then on the next day, at least how we do it, we have again another solution, a so-called loading and cryoprotecting solution. They differentiate a bit in the chemical, but basically it's again sucrose and glycerol. So the glycerol goes quickly into the tissue, can stabilize again the membranes, and the sugar probably follows. And they again take the water out and they form a mass which is extremely thick and has extreme low water content. The shoot tips probably do not survive this procedure for a long time. That's very limited in time where you can apply this procedure. But if you have this thick mass these components and throw it, let's say, into liquid nitrogen, you have then immediately this vitrification process. That means they have not the chance to produce any ice crystal. They just stop everything and freeze. It's like an extreme thick glass. Therefore, the name comes from vitrification, is the glass transition. And glass means that it's a physical property where you still have a bit of movement, but most likely not. But they are also not able to do anything else, let's say. And when you have this material then in the liquid nitrogen, it's stable. It's assumed that they do not metabolize at all. However, we cannot measure this really at minus 196 degrees, but probably any reaction, they are extremely low, that they are storable for a long time, let's say. Yeah, and then when they come back, so you heard already from me, we throw it into liquid nitrogen. So they have freezing rate of about 1,000 degrees or more. So it's extremely fast. And to avoid for the ice crystals, also the heating process, the warming, we say, this needs to also very fast. But here we have, of course, a biological limitation because, proteins denaturate at 70 degrees. So we can just go up to 40 degrees to avoid any problems, any damage to the tissue. And therefore, the warming rate is not as fast as it sometimes might be. And this is also a limiting factor. However, in potato, it works quite well. We do that, we put that in 40 degrees for a minute, and then it goes on a tissue culture medium, and then they really start to grow. So, and that's the fascinating thing. They can survive this vitrification approach and are able to generate at a very high percentage also. So that's the vitrification approach in principle. Of course, you have also other approaches. They are, for example, fruit trees. They are also species which are cryopreserved. Not here, but in principle, apple or pear are the species which are in cryo, and they have a slightly different approach. They put it into slow freezing. And this means that you have them in a cryoprotection. I'm not so extremely sure about that, but you put them in a freezer where you have a low cooling rate, just 10 degrees per hour, something like that. And you go up to minus 50 degrees. And the idea is that you slowly take out the water by this approach, and then you throw them into liquid nitrogen. This goes also back to the vitrification then at the end, but the procedure is different.
I think it's really, really incredible to hear about the complications of that process, especially all of the great troubles to make sure that water stays out of the plants at all times and doesn't create the ice crystals. And I think it's enormously fascinating to think about how many scientists must have been working for decades to get this process working as it is now to be more efficient. And I think that after understanding the science a little bit, it gives me a little bit more appreciation into one other area I wanted to ask about, which was the entire economics of the situation. Because I saw one of your recent papers talking about the different kinds of cooling mechanisms that we might have and the cost therein. And that's when I paused to think about how enormous the cost might be to not only have all of this specialized equipment, but also take all this time to do this painstaking process, but then also have so many of highly educated people in the agricultural industry working on this. So the two questions I had were, what's some of the major costs of this process? And then who is most, say, economically incentivized to go out and fund this type of thing?
Also, very important questions. Just to give you an idea, so at the moment, we have estimated that there are 400,000 crops in the field gene banks. Of this 400,000 crops, there are about 40,000, roughly, let's say, are in the tissue culture, are somehow back up. Sometimes you can back up them also in different fields, but anyway, they are somehow back up in the tissue culture, and only about 10,000 are in cryopreservation. And as I said, the cryopreservation is the only approach to maintain the species on a long-term run. Because in the field and in the tissue culture, you have also the great risk that this material develops mutations. You cannot avoid that. That's a natural process. It's pushed by just microbes or just by changes in the environment, by stress. This is really a natural process. And to keep this plant as it is, cryopreservation is the only one, because here you have the material without any metabolic active process. So this should ratify why we do that. And together with other gene banks, we really think how to get this huge amount of plants in the field gene banks into cryo, because we have a great risk, especially with environmental change, that we lose a lot of this collection. And this includes very important fruits like cacao, coffee. You have major fruits in Brazil. This is cassava, this is yam, for example, where really huge amount of people depend on. And this is very important that we have them somehow saved and back up in a gene bank. But as you said, you need highly dedicated people, you need very specific structure and facilities, and it costs a lot of money. The biggest cost is to bring this material into cryo. Of course, because you need these tissue culture facilities, you need all the equipment, but as soon as you have it in cryo, then the costs go down because you just need liquid nitrogen. Of course, this is very dependent on the area where you are. When you are, for example, in Germany and we have this high personal, the staff cost, then of course cryopreservation is extremely high, but also the field maintenance is here high because of this cost of the staff. When you look at, just an example, African countries, South American countries where the staff cost might be a bit lower, then of course also cryopreservation might be lower. But usually then this is not done there. Just to give you an idea, we have done, my colleague, he has done a comparison between costs of the field and of the cryopreservation. And after about 15 to 20 years, the cost for cryopreservation become neutral, because you have a permanent cost in the field of every year working on the plants, transferring from one field to another, whereas in the cryopreservation, after you brought it into cryo, you just have the liquid nitrogen. And this is dependent, of course, on different factors. But if you look into other centers who do cryopreservation, they have similar numbers. But of course, this differs really on the region where you are. To get this funding, this is extremely difficult. I'm happy that Germany pays this. So we are, as said, I'm in the Gene Bank for Horticulture and Agricultural Plants here in Germany, and we have our own unit to do this kind of work and to bring our potato and all the vegetables, all the plants which need to be vegetatively propagated into cryo. However, I still have a lot of work. But worldwide, the situation is a bit critical. It was a big and good thing that in the 60s, the FAO established the Institute for Plant Genetic Resources in Rome. And they started to set up and they established different gene banks. And they are huge centers, around 7 huge centers worldwide. They focus on specific crops. Among those is the SIP, for example, in Peru, which focus on potato. And there's also biodiversity in Leuven in Belgium. They focus on banana, for example. And these are the crops which also need cryopreservation. And there is at the moment so-called the crop trust behind. And they try to organize the money for these centers. This money comes from World Bank, from international funding associations like here this Melinda and Bill Gates Foundation. It comes also from rich states itself, and they try to get the money to have this approach also used and to really back up these resources which are otherwise are lost.
Yeah, and I think that, as you said, this area of research is going to become more and more important in the future. And regarding that, I just wanted to wrap up with two rapid fire questions about the future of this discipline. So the first being, given all of these incredible uses of plants, but also the increasing challenges that we're seeing in maintaining the diversity, how do you think this field is going to be changing in the coming years?
Oh, yeah, that's a good question. I think at the moment, I don't see a change. I think we just need to focus to really bring all these plants into cryopreservation. But I'm not sure if I can see the whole picture. I mean, in terms of all these breeding approaches we have, I mean, our resources are essential for future breeding approach, and it goes more and more in the direction that we try not to modify the species in a way that we cross, I don't know, dog with wheat. We would like to stay within the species or within the genus, and then you have maybe white wheat with the cultivated wheat, and you still need to do something because it doesn't work naturally. Therefore, you need these resources, and sometimes they are only available in cryopreservation because otherwise it gets lost. And for us, our focus is at the moment to really to store them as soon as possible and with the best efficient approach, because it can be so quickly that you have a genetic resource lost. And I have to tell you, we do a genetic study and collected leaf material from our garlic species in the last year. And this year we decided we will make available the dry leaves also for other users who can use that for genetic studies. And during we did that, we realized, oh, we don't have the plant anymore in the field. What was very surprising and actually also frightening for me, how quickly it can come that we lose such accessions. I mean, this happens in the gene bank, but I think this happens in the nature, in the agriculture, even at a higher level. So at the moment, our focus is just to do cryopreserve as soon as possible with the best approaches.
I think that's a very interesting perspective from the people at the definite edge of the industry, because at the end of the day, it is what your priorities are that are going to be communicated to other stakeholders and to hopefully the people who are willing to fund this. So hopefully we get to see more of that change soon. And finally, if anyone's more interested to learn about this area and your work, where might they go?
Of course, they can check our webpage of the IPK, of the Leibniz Institute for Plant Genetic and Crop Plant Research. And we try to set up shortly, but it's not at the moment, a website about plant biobanking. But this will come up soon. We also produce some YouTube videos, but I think that's at the moment future stuff. But I think this community will be available if you look for the keywords plant biobanking and cryopreservation.
All right, so I think that'll be great for anyone who's more curious about this area. And thank you again for taking the time to walk us through everything from the history to the science to the economics and the future now of this incredible area of research.
Okay, thank you very much also for your interest, and I hope I could help you a bit in the understanding and that you also get excited with this very nice field.